Members of the Congress, hello from Australia. I thank the organisers at LAPAN and UFMG for inviting me to speak at their 10th Brazilian Congress on Vision Neurosciences. Congratulations on your 10-year anniversary since the first Neurovision Congress. I'd like to talk about this study. I conducted it several years ago, but I decided to present it for two reasons. One, found preliminary evidence of potential genetic marker for the visual disorder Mears-Urlain syndrome, nowadays also known as visual stress. The other reason I chose this topic is because the paper was published in the very same year as the first Neurovision Congress. So another anniversary of sorts. This short excerpt from the abstract basically says that in a gene called ApoB, normally associated with cholesterol levels, we found differences in the alleles or variants of this gene in a small group of visual stress patients as compared to the alleles carried by the control group. What led me to this study? Well, back then in 2012, I was well aware that many studies had published evidence of abnormal lipid metabolism, not only in Mears-Urlain syndrome or visual stress, but also in dyslexia, ADHD, chronic fatigue syndrome, and even in mild autism, autistic spectrum disorder. What interested me was the fact that the symptoms of visual stress had been reported in all of these other conditions as well. So I then realised that several studies had found in visual stress patients unusually low levels of cholesterol, particularly the form of cholesterol known as LDL, which is often called the bad cholesterol because high levels of that form of cholesterol can lead to cardiovascular disease, heart attacks, strokes, etc. Now, one or two studies that also found this in dyslexia and ADHD. But more than these three studies shown here, had found low cholesterol levels in visual stress and also chronic fatigue syndrome. And interestingly, this study here by Sparks et al. At the University of Newcastle, Australia, found that over 50% of their chronic fatigue syndrome patients were also comorbid for visual stress. So I began to think that a genetic study of a gene called ApoB in visual stress patients would be interesting because Firstly, LDL cholesterol is primarily carried through the body by being bound to a lipoprotein known as Apolipoprotein-B. And the ApoB gene codes for production of this lipoprotein. This is why ApoB has been linked to both high and low cholesterol levels. And what makes this particular gene one of the most widely studied genes in biomedical circles is because it is highly polymorphic. It has many variant alleles. Now, I should explain, alleles are simply slightly different versions of a gene. Every gene has a few different versions or alleles that vary from each other. For example, the gene for eye color has 3 or 4 different alleles. And we inherit one copy of each gene, one allele of each gene from each parent. So we have 2 copies. With eye color, for example, if we have one allele for that gene for brown eyes and another allele for green eyes, for example, then we end up with a color in between somewhere. However, and most genes only have a few alleles, but the ApoB gene in populations as small as 100 people has been found to have at least 17 variant alleles, which makes it ideal for study, and that is also why it is used in practical experiments in universities for students learning to use biotechnology equipment and so on. So I approached my university about a genetic study of the ApoB gene in visual stress individuals, and my supervisor said, for ethics approval of this genetic study, you'll have to show me evidence that cholesterol is actually important for vision. A quick review of the literature showed that cholesterol is not only important in vision, it is crucial for optimal visual function in many ways. Here are only 3 examples. Cholesterol, or particularly LDL cholesterol, is the only molecule that can transport essential fatty acids, which are also lipids, through the blood-brain barrier into the retina and also into the brain. Now, all cells need essential fatty acids, hence the name essential. However, neurons need large amounts of essential fatty acids, and the retina particularly needs the essential acid DHA. Without cholesterol, there would be no essential fatty acids in the retina or the brain. Now, another important role for cholesterol in vision is it actually puts a limit on the amount of activation of the photopigment rhodopsin. Rhodopsin is a retinal pigment that's found in the rod cell photoreceptors. Rhodopsin is responsible for capturing the energy of photons of light and converting that energy into chemical energy. And this initiates a chain of chemical signaling known as the phototransduction cascade, which is responsible for the visual signal transmissions through the optic nerve to the visual cortex. The trouble is, well, the problem for rhodopsin is that all photons are not created equal. Photons of short wavelength light, such as blue and violet, have far more energy than photons of yellow and red light. And thus, when exposed to bright light with blue light content, such as when outdoors or under bright fluorescent lighting, the rhodopsin becomes overactivated. And were it not for cholesterol putting a limit on the amount of activation, the signaling from the rod cell photoreceptors to the visual cortex would be enormous and would overwhelm the visual cortex, or at very least cause extreme visual discomfort. And that is the reason that cholesterol has also been found to reduce visual discomfort to short wavelength light. Interestingly, visual discomfort to short wavelength light is one of the most common symptoms of visual stress. Lastly, the phototransduction cascade itself is also regulated by cholesterol again. Permission was granted for a small genetic study of the ApoB gene in a group of visual stress patients and a few controls. And before I move to the experiment, I should show you where this gene is located. It is on chromosome 2 on the short arm of this chromosome. This is the long arm, and all 23 of our chromosomes have a short arm and a long arm. Of course, we have 2 copies of each chromosome, one from each parent, but we have 23 different types of chromosomes. Now, what added to my interest in this gene was that when I looked at the location, I found that a gene called DYX3 is a close neighbour to the ApoB gene. And DYX3 is a gene that is known to confer the visual what should I say? Around the year 2000, it was hailed as a gene for dyslexia, because at least one variant of this gene, one allele, if inherited, makes a person much more likely to have reading difficulties and therefore dyslexia. However, people with visual stress also have reading difficulties and have been misdiagnosed with dyslexia. And lastly, another coincidence, virtually next door, it's as though these 3 genes are in the entire human genome. These 3 genes are all living in the same street, which I think is a coincidence, because this 3rd gene is associated with chronic fatigue syndrome symptoms. This is not the final experiment. It is the 4th trial of a technique called electrophoresis that we used with only 8 subjects. We wanted to make sure we had the methodology perfectly refined. Now, this is how scientists can actually visualize a gene of interest and see if it has different alleles, or if the person carrying it has different alleles, or if they're carrying identical alleles, their DNA sample of this gene will appear as one bar in this photo. If they've inherited two different versions of the gene, there will be two bars. The reason that this gel is this simple device is so effective is simply a tray, small tray, this big plastic tray filled with a thick, almost solid, transparent gel, a clear jelly, in other words. And to visualize a gene of interest, DNA samples are given by the participants. The gene of interest is isolated, and the DNA only from that gene is placed into each of these wells on one side of the gel. Now, this diagram here, we only used 8 individuals in the first 4 trials. Here they are here. These 2 lanes and these 2 lanes are genetic material that comes from the manufacturer of these kits. These are simply 2 identical. In these lanes here, there are 2 identical fragments. This is to make sure that they travelled, the experiment worked equally on both sides of the plate. I should have explained that first, that after the gene samples are placed for each individual as an individual lane, after they are placed into the wells, a small electric current is turned on, 50 volts. And because the positive anode runs along the other side of the plate and DNA is slightly negative, the molecules are attracted to the other side of the gel. Now, eventually, they will all reach the other side and there'll be nothing to extinguish. So therefore, the power's turned off after a given time. And then, using ultraviolet light, a photo is taken, which gives us a snapshot of how far they progressed through the gel in that time. How this separates the alleles is different versions of a gene or different alleles have slightly longer or slightly shorter molecules of DNA. And this gives the smaller molecules an advantage to get through the thick gel. They can pass through this thick, almost solid gel more easily than slightly longer samples of DNA. So if a person has 2 alleles that are different, they will travel 2 different distances and we can see them. Now, on both sides here, this is how we estimate the length of the alleles looked at in this experiment, because this is known as a DNA ladder. It's a sample that goes into any well. In this case, we had so much room we put one each side. And it contains 20 or more fragments of DNA of an exact length, precisely known length. And the length of each of these fragments of DNA, the difference between them is identical, 100 base pairs, which is a unit of measure of how long a DNA molecule is. So these shorter molecules on this ladder travel further, and the distance between them is always the same, though, as far as the length of the molecule. So, for example, if this marker here on the ladder is 600 base pairs and this one is 700 base pairs, then we know that the DNA, well, this is the sample, but the DNA in most of the individuals here were somewhere between 600 and 700 base pairs long. And we can refine that further to get a reasonably accurate estimation of the length of the molecule. Now, I'm talking way too long. So that was successful. I thought I'd show you that first because the final experiment we did with the full group, a pilot study, so only 10 individuals with visual stress and 8 controls, it's not quite as clear. It's clear on a biotechnology laboratory computer designed for this, but it's not so clear on my laptop. In this case, there were 18 different individuals, and we put the marker, the DNA ladder marker, in lane number 11. Could have gone anywhere, but I suggested putting it in lane 11 so it's easy to see that the 10 lanes here are the visual stress subjects and the 8 lanes here are the control subjects. And we found differences. First of all, 4 of the 8 control subjects were homozygous. That means they had identical copies, identical alleles for the ApoB gene. And the other 4 were heterozygous. They had 2 copies, which are clearly seen here, and especially here. Two variant alleles of ApoB, I should say. Now, with the visual stress group, all 10 individuals were heterozygous, 2 different alleles of ApoB. This was the first finding, and it was significant because it's unusual to find so many heterozygotes and all of them in one group. The other finding is that we found differences in the mean lengths of the DNA fragments in the control group compared to the visual stress group, using this ladder as for calibration. Because these had travelled further in the same time, the alleles of the control group are shorter. In fact, they were 13% shorter than in the mean length of the alleles of the visual stress people. And before I move to the results, I should point out that because this was my first genetic study, I had this photo on the wall in my house, and when friends who are not scientists would visit, they would ask what it was. I would explain, these are the visual stress people and these are the control people. Can you see any difference between the groups? And almost everybody said the same thing. They said, I know nothing about this, but it looks to me like these guys in this group are further down the photo than these guys in this group. And that basically is a key finding of our study, actually. So here are the results. This will make it much easier. We found 11 different alleles, variant alleles, of ApoB in the 18 subjects or participants. We named them allele variant, AV1 to AV11. And here they are here. These are the 11 alleles. As you can see, from these 11 alleles, the white columns represent the number of those alleles in the visual stress group, and the black columns represent the number of each allele, or the frequency, percentage of individuals who had that allele in the control group. And as you can see, there's an entirely distribution of the 11 possible alleles of ApoB between the 2 groups. A key finding is that in the visual stress group, allele 1, 2, 7, 8, 10, and 11. All of those alleles were not carried by anyone in the control group. Admittedly, only 8 controls, but it's unusual that none of these alleles were in the 8 controls, because every person carries 2 alleles, whether they're identical or not. So the control group had 16 alleles, and the 10 visual stress patients have had 20 alleles, 2 each, one from each parent, sometimes identical versions, sometimes different versions of the same gene. Basically, the last thing I'll say about this before I've moved to the end of this presentation is that with only 10 subjects, you can already see the beginnings of a nice bell curve, which is found in population studies. And if we'd had more participants, we may have had this might have been the beginnings of a bell curve of the frequency of it in the control group. So the 2 groups have basically some overlap, but they tend to carry different alleles of the ApoB gene. That is the number one finding. The key findings, as I said, were the mean length of the alleles in the visual stress group were 13% longer than in the control group. 4 homozygous from 18 participants were in the control group, and none were in the visual stress group. This was statistically significant. In the control group, these 2 alleles were very common, but not in the visual stress group. And most importantly, I think this is the main finding of the study, of the 20 alleles carried by the 10 visual stress participants, remember each person has carries 2 alleles, 14 of these, 70%, were not carried in any one of the controls. There was not a single control who carried any of the 14 alleles, these 14 alleles carried by the visual stress group. I always thought for a large study with many visual stress participants, we could possibly enlist these visual stress participants from Mees-Erling syndrome diagnostic centres. If not, a study simply looking at the cholesterol levels of a large group of individuals with visual stress would be very interesting. And further in the future, these 2 points are far more important, but in the future, it would also be nice to examine the ApoB gene, ADHD subjects, both with and without visual stress symptoms. And lastly, I think it would be very worthwhile to investigate in the general population, to investigate if low cholesterol levels in general might actually correlate with cases of visual discomfort and diagnosed reading skills problems in the wider population, not only in visual stress or ADHD. It's just a hunch or intuition I have that I think we may find that low cholesterol and visual discomfort are closely associated. So thank you for your interest, and again, congratulations to the organisers on their 10th Neurovision Congress. Until the 11th Congress next year, bye-bye.